everyone. I'm Vineet from Benchling, and I'm an engineering manager here. For those of you who haven't heard about Benchling, we're an Andreessen Horowitz-backed startup based here in San Francisco, and our mission is to accelerate the pace of life science research. Today I'm going to be talking about scaling the new gene editing technique, CRISPR, and how we used AWS Lambda to do that. Now, before I get into that, let's talk a little bit about Benchling. Benchling was founded in 2012 out of MIT. And we've come a long way since. We have over 70 enterprise deployments, a presence at several of the top 10 pharma companies, and over 130,000 scientists using us worldwide. Now, what we're trying to build is a platform to unify all the different types of data that scientists are using in a lab. And on top of this platform, we have a number of different custom applications, from our molecular biology suite, which allows people to work with DNA and protein sequences, to sample tracking for people who are working with containers and plates in their lab. And we think by unifying all of this data and making it easy to use and query, we can streamline the way research is done and make it get done a little faster. As we heard about in the keynote, iteration speed is really important. And we think by making iteration speed faster, we can affect how fast progress is done in science. How long does it take you to go through a trial and error workflow? How long does it take you to test a hypothesis? This isn't true just for science. This is true in basically every domain. Let's take programming as an example. How long does it take you to run your tests? How long does it take you to refresh your page when you're building that new front-end component or deploy your code to production? Now, all of these things take minutes, if not seconds. But your developers are still going to complain if they're a little bit too slow. If your front-end page takes 5 seconds to reload instead of 1 second, because it affects how fast they can work. But now imagine this as a biologist. If you want to grow a new cell culture, that's going to take you 3 days. If you want to express a new protein, you're going to be waiting 2 weeks for that to happen. Now you're growing a new strain of mice. You need to wait 8 weeks before those mice are going to grow up. Now, we're not in the business of making mice grow up faster or cell cultures grow up faster. But what we can do is prevent you from making mistakes. We can make sure that you're not going to spend 8 weeks needlessly when you need to run your experiment again. And we can do that using software. So today what I'm going to talk about is how we did that for CRISPR. How many of you guys have heard of CRISPR before? All right, basically the entire room. So I'm going to talk a little bit about the history of gene editing in CRISPR, and then dive into the meat of this talk, which will be the problem we're actually solving and the algorithm and infrastructure that we use to solve that. So let's start with gene editing, and start with some brief vocabulary. You all are familiar with this picture, the double helix. This is DNA, the basic building block of life. It controls everything that happens inside your body. Now, DNA is just a long string of nucleotides or bases, or in programming terms, a long string of characters. And this DNA makes up what are called genes. These genes in your body have various functions, from the color of your hair, to how good your eyesight is, to whether or not you have Huntington's disease. And your genome is just the collection of all the DNA in a single organism, like a human. So gene editing is exactly what it sounds like. Can we actually make edits to these genes to add functionality to that, to remove functionality, to turn it on or off? So let's go through maybe a specific example. So one way you can use gene editing is to just completely break a gene or turn it off. Now, why would you want to do that? Well, maybe I don't know what the HTT gene does. So maybe I turn it off, I test it in some bacteria, and I see what happens. I can try and figure out what that gene is actually doing. In this case, the HTT gene is actually the Huntington gene, and maybe you have a mutation in that. And maybe by turning that gene off, we can actually prevent you from getting Huntington disease when you get to 35. So this is just one application of gene editing, and there's a lot of different applications. But gene editing existed before CRISPR. People have been doing this since 1991. And the problem was that not that it didn't work, the problem was that it was just too slow. It would take months to actually make these gene edits. There were entire PhD projects of people trying to make these edits until CRISPR came out in 2012. And this completely revolutionized how people were doing gene editing, because now you could do this in days. A high school student could get trained, and they could order a kit from a company and start using CRISPR immediately. And that completely changes the iteration speed that these scientists are working at. Now, unfortunately, software didn't quite catch up. So this came out in 2012, and in 2014, scientists were starting to use CRISPR. But even in 2014, actually planning these experiments was taking hours or days. Some of our customers mentioned it would take weeks to actually plan out a CRISPR experiment. Now, why do you actually need software to do this? Well, let's take a look at a more realistic example. Instead of just one gene, actually, before that, let's talk about how CRISPR actually works. So as I mentioned, we're trying to break a gene here. So how does these scissors actually know where to go in the genome to break? Well, we guide those scissors using what's called a guide. This guide is just a short piece of DNA, maybe 20 characters long. And that guide is going to find a region in the genome that's a match to that guide. And the scissors are going to make a cut there. And if that cut happens to be in a location that's in a gene, we can break that gene. Well, let's take a look at a more realistic example of why we actually need software to do this. Let's say I have two genes here, and I'm trying to target this HTT gene. And in this toy example, I'm using a 4-character guide of AGCT. So we're going to guide our CRISPR there. It's going to make a cut, and maybe everything will work fine. Unfortunately, I didn't notice that there's also this AGCT somewhere else in my genome, and we accidentally made a cut in this BRCA2 gene. And unfortunately for me, that's actually a tumor suppressor gene, and that prevents things like breast cancer. So when we're using CRISPR, we need to make sure that we're targeting the regions that we care about and not targeting anything else. And this is where software comes in. We can find guides that match the region and the genes that you care about, and specifically don't match anywhere else in your genome. Now, back in 2014, the software solutions to do this were taking hours or days. And we saw this as an opportunity to help science move a little bit faster. So now we have a problem statement. A scientist is going to come to Benchling, and they're going to ask, how do we knock out this particular gene? And we know what they mean now. They're trying to target usually the beginning of that gene to break it. And we want to find guides that match that region and don't match anywhere else in the genome. So how does Benchling actually answer this? Well, we find a bunch of potential guides in this region, and then we test all of those guides against the rest of the genome to see, do any of these guides cut anywhere else? Most of them probably aren't going to work, and we're going to find the best candidates for those to present those to the user. Now, what does match actually mean here? I've been saying the guide needs to match in this region and not match somewhere else. Well, it turns out we're not looking just for exact matches. CRISPR might cut even if these guides are close. And empirically, CRISPR might cut up to an edit distance of 4. So in this case, the guide might match this location in the genome, even with just 3 mismatches here. So this is a little bit harder of a search problem. We're not just looking for exact matches. We're looking for approximate matches. So let's phrase this more as an algorithmic problem. Given a genome and a set of maybe 200 guides, we want to find all of the locations in the genome that actually are within an edit distance of 4 of one of these guides. Now, this isn't just a theoretical problem. This is a real problem we need to solve. So what kind of numbers are we looking at? Well, the human genome is around 3 billion bases long, and that's a longer end of the genomes. We want to target a guide length of around 20, and typically we're going to be looking at around 200 guides at a time. Remember, most of these guides aren't going to work, so we're going to test a bunch of them to try and find some that work for our use case. So as we're building out this infrastructure, what kind of goals are we trying to hit? We want this to be fast. Remember I said this was taking hours or days to compute before? Well, we have a web application. We want people to be able to click a button, say run a CRISPR analysis, and get them a response immediately. Second thing is we want this to be scalable. There's going to be millions of searches a month. And we also want it to be cheap. We're a startup, as many of you are, and we care that this isn't affecting our bottom line too much. All right, so now let's get into the fun part, the actual algorithm and infrastructure that we use to solve this. So naively, we have a pretty straightforward search problem. We can iterate over the entire genome, and for every location, maybe we can find whether it's a match or not, and we can do that every time. And that sounds like a pretty parallelizable problem. We could divide this genome into several chunks and do them all at once. On the other hand, we could index the genome. We could store some sort of queryable format so that when queries come in, we can answer those even faster. So let's go through some possible solutions here. Then the naive approach here, you can take a look at the code I have up here, but just for every guide, go through the entire genome, check the edit distance between my guide and that location in the genome. If the edit distance is less than 4, we found a match. Great. Turns out this actually isn't that slow. It takes 3.5 hours. But if we're running this 1 million times a month, 3.5 hours is going to be really expensive. So how can we make this a little bit faster? Well, we were iterating over the genome 200 times before, so maybe we can find a way to do that a little bit less. Remember, this genome is 3 billion characters long. So instead, let's take our input 200 guides and find every possible 20-character string that's within an edit distance of 4. We'll find all of our potential matches all up at the beginning. So there's 425,000 or so possible matches for any particular guide. So we're going to end up with 85 million possible matches at the beginning. But now we can just iterate it over the genome once. We can take a given location, check if it's in our giant hash set. If it is, we found a match. But now we're only iterating once, and we have a solution that only takes 1 hour. It needs around 2 gigs of memory if we're going to store these 85 million strings. Not too bad. Again, 1 hour is still pretty slow. It's going to cost us a lot every month. And it's still not really parallelizable down to 1 second. It's still a little bit too slow. But we have a couple of other tricks up our sleeve. The first one is we know what space we're in. We're working with DNA. And DNA is only 4 possible characters. We can represent each of these characters instead of as a letter as 2 bits. We can map, in this case, A to 0, 0. And we can convert our 20-character guide into just 40 bits. Now I can represent those 40 bits in a 64-bit integer, with 24 bits to spare, in fact. Now, why is that helpful? Well, instead of working with 20-character strings, I can now work with 64-bit integers, and that's going to make a lot of things faster. Before I had a hash set of 85 million 20-character strings. Now this hash set is a lot smaller. Hashing on these integers is going to be a little bit faster than on those strings. As we're iterating over the genome, instead of keeping track of 20-character string of where I am and appending and prepending to that string, I can actually use bit shifting operations on my 64-bit integer, or what's called a rolling hash, as I'm going through, and that's going to be a lot faster. In this case, it turns out to be 5 times faster, and we're all the way down to 13 minutes, and using a lot less memory because integers are smaller. 13 minutes isn't bad. It's still going to cost us a little bit more than we were comfortable with. So what else can we do? Well, it turns out that when you're iterating over a 3 billion character string and checking existence in a hash set, hash sets end up being pretty expensive. Just doing the hashing operation takes a long time if you're doing it 3 billion times over and over again. So how can we avoid that? Well, maybe we can represent the hash set as an array instead. So we take our hash set of 20-character guides. Now remember, we're representing these as integers now. So in this case, let's imagine that I have a hash set of 1, 3, 5, and 6. Now I can represent that as a Boolean array instead, where the value is true if that index is in the hash set. So in this case, the largest element is 6, so our array is of size 6. This ends up being a lot faster. I can do an array lookup instead of hashing the integer and then checking the existence in the hash set. But if this is so much faster, why doesn't everybody use Boolean arrays instead of hash sets? Well, it turns out it has a couple of problems. One, we need to have integer keys in the first place. In this case, we do, so that's fine. The major problem, though, is the size of our hash set needs to be as large as the largest possible element. And in this case, it's just 6, so we have an array of size 6. But if I had an element here that was 10 million, my array would now need to be 10 million elements long. For our problem, what's the maximum possible number? Well, we have 4 characters at each position. We have 20 characters. So we're going to need 4 to the 20 spaces in our array, or 1 trillion elements in our array. That's quite a lot. We're going to need a terabyte of memory to run this algorithm. And even just making those memory accesses is not going to be slow because it can't fit in cache. But how can we use this approach to use array lookups instead of hash sets? Well, what if we split it into 2 arrays instead of 1 array? So we started with this hash set. We have these 64-bit integers. Each of these is representing a 20-character string. Now to split it into 2, I'm just going to split every string into 2, take the first half, put it in hash set number 1, and take the second half and put it into hash set number 2. So in this case, the first, here I'm using 8 characters, but you can pretend that this was 20. You see the CGTA in the first hash set. You see the GCTA in the second hash set. OK, so we split 1 hash set into 2. Why is that helpful? Well, before, the size of our array needed to be 4 to the 20, because there were 20 characters in each string. Now, our array only needs to be 4 to the 10, because there's only 10 characters in each string. So we've now dropped from 1 trillion elements in our array to 2 arrays that just need 1 million elements, just 1 megabyte each for each of those arrays. So that's a massive improvement in memory. How do we actually do a query? Well, let's say this was our query at the bottom. I take the input string, I divide it into 2, check if the first half exists in my first hash set, check if the second half exists in my second hash set. If it does, great, we found a match. Again, this seems too good to be true. What's the catch? As I'm sure many of you guys have noticed, we might need to deal with false positives here. I can mix and match the first half of one string with the second half of a different string, and now we might think there's a match when there's actually not. It turns out that false positives is actually a real problem we need to deal with. I don't have as much time to talk about the tricks there. But basically, the summary is instead of representing this as a Boolean array, we can represent this as an integer array. And that integer is now the edit distance between that half of the guide and the closest guide in our original set. So instead of checking if both Booleans were true, we can add up the two edit distances. If those are greater than 4, we know this is not a match. And we actually avoided 50x of our false positives and got this algorithm all the way down to 8 seconds. This is a 100x improvement from where we were before. Now this, at the surface, is somewhat incredible. We're iterating over a 3 billion character string, searching for 200 guides, and we can compute this serially in 8 seconds with just 2 megabytes of memory. And this is just our iterative solution. And we can parallelize this if we want to. We can split up a genome into 2 or 4 and make this 4 seconds, 2 seconds, and we can get it all the way down to 1 second. Now, this is the iterative solution. Can we do better with our indexing solution? Well, we know that each genome is going to be around 3 billion bases. We want to be able to support around 1,000 genomes. We're not just dealing with human genomes, we're dealing with plant genomes, bacterial genomes, animal genomes. So this is going to be just 3 terabytes at the start. And we played around with some simple indexing solutions that we could use, and anything even basic required at least 30 terabytes of space. We actually weren't able to come up with any indexing solution that was faster than our iterative solution and used less than several petabytes of space. And even at 30 terabytes, the cost is going to be somewhat of a factor here. So let's talk about cost a little bit. So before I get into cost, let's figure out the infrastructure so we can actually analyze that. For our iterative solution, it's pretty simple, nothing crazy here. We have a task come in, a genome, and some guides. We put it into an EC2 instance. That instance decides how many times we want to parallelize this. We know it's going to take 8 seconds, so maybe we parallelize it 8 times. So we send those to different worker instances who process those particular regions of a genome. They download that part of the genome from S3. They do an analysis. They return the response. We now have a solution that works, and we actually used this architecture for about a year. And this ran in under a second. But we noticed some problems with cost, and this is nothing different than most web applications, where we were paying for time for these EC2 instances when we weren't really using them, nights, weekends, during the day. So what's the common solution for this? Well, let's throw an autoscaling group on these worker instances. Without autoscaling groups, this ends up costing maybe 30K a year. We only need around 10 instances to support what we need. With autoscaling groups, this cuts down in half to around 15K. But perfect autoscaling is really hard here. We can solve this problem on nights and weekends, but during the day, 54 seconds out of every minute we might not be doing a CRISPR analysis, but we're still going to be paying for that time. So, as I mentioned at the beginning of the talk, instead of using autoscaling groups, what if we replaced these workers with Lambda functions? I can define a function that just runs our algorithm. It takes a part of the genome to analyze, downloads that from S3, runs the algorithm, returns the response. This is basically just a drop-in replacement for what we were using EC2 before, but it actually solves all of our autoscaling problems. We no longer need to pay for time that we're not really using. We don't need to maintain an EC2 fleet anymore. And this is going to end up costing a lot less money for us. Let's take a look at the actual costs here. So I mentioned we couldn't come up with an indexing solution, but let's pretend that we did. Let's pretend that we actually missed a solution here, and there was some indexing solution that is so fast that we wouldn't need to pay for compute at all. So we're just paying for storage. Well, at 30 terabytes, this is going to come in at around 100K per year. Now that's pretty expensive, and we need to store that in some queryable format, maybe Dynamo or RDS or something. But that's pretty expensive. So let's compare that to our EC2 solution here, which was only costing 15K per year, that we were mainly paying for compute. But now when we switch to Lambda, we cut that by a factor of 5, because we're only paying for the time that we're using. And now we're just paying 3K per year to run 1 million searches of CRISPR a month. Even better, we don't need to manage an EC2 fleet, so not only is it cheaper, it's also less effort from our developers. So going back to our original goals, we now have an algorithm that's 8 seconds that we can parallelize to basically as fast as we need. Is it scalable? Well, we're using Lambda. It's about as scalable as we can get. And lastly, it's cheap. We're paying 3K a year. It's at the point where it doesn't make sense to invest any more time trying to make it faster, because we're not really paying any money, and we can already make it as fast as we want. So CRISPR is just one example of technology that scientists are using today. But we felt that this was an opportunity for us to use our software skills and use technology to help move science forward. But we're also really excited about all of the new technologies that are coming out in science, from gene therapy to cell therapy, antibody engineering, synthetic biology. All of these areas have a lot of innovation going on in them, and we are really excited to help. We're excited to figure out how we can use our technology to make these move a little bit faster, to help them draw conclusions faster, to help them go to market with their drugs to cure cancer and cure Huntington's disease a little bit faster. Let me know if you have any questions. I'm happy to talk about any of this, or if you're interested in working on any of these problems. We're really excited about the future of biology, and our mission is to accelerate the pace of this research over the next 10 years. Thank you.